DNA Structure and Replication. Journey into molecular biology. Welcome to our video on DNA structure and replication. Today, we will explore the amazing world of DNA, the molecule that carries genetic information in all living things. Let's get started. Let's begin with understanding the structure of DNA. DNA looks like a twisted ladder and has two strands. Each strand is made up of sugar and phosphate molecules connected by steps called base pairs. The base pairs are like the rungs of the ladder, and they always match up in a specific way. Adenine matches with thymine, and cytosine matches with guanine. These pairs stick together with weak bonds, holding the ladder structure in place. It's important to know that the two strands of DNA run in opposite directions. One strand goes from the 5' end to the 3' end, while the other goes from the 3' end to the 5' end. Now, let's talk about DNA replication. This process is essential for cell division and passing on genetic information. During replication, each new DNA molecule has one old strand and one new strand. DNA replication happens in three main steps: initiation, elongation, and termination. Initiation. In initiation, special proteins unzip the DNA, creating a fork where the strands separate. This exposes the individual strands for replication. Elongation. During elongation, building blocks called nucleotides attach to the exposed strands. These nucleotides match up with the existing strands, creating new matching strands. One strand is made continuously, while the other is built in small pieces. Termination. In termination, the replication process finishes as the new strands are complete. The result is two identical DNA molecules, ready to carry genetic information. This diagram is showing parental DNA strand where DNA replication takes place. First, helicase unwind the parental double helix. Then, single-strand binding proteins stabilize the unwound parental DNA. After that, the leading strand is synthesized continuously in the 5-3 direction by DNA polymerase. The leading strand is synthesized continuously while the lagging strand is synthesized discontinuously. Primase synthesizes a short RNA primer, which is extended by DNA polymerase to form an Okazaki fragment. After the RNA primer is replaced by DNA by another DNA polymerase. DNA ligase joins the Okazaki fragment to the growing strands. Proofreading and error correction. To ensure accuracy, special enzymes in the cell can detect and fix any mistakes made during replication. This helps maintain the integrity of the genetic information. During DNA replication, helicase unwinds the DNA strands. DNA polymerase builds new DNA strands using existing strands as templates. DNA ligase joins the newly synthesized DNA fragments. Exonuclease proofreads and corrects mistakes. Real-life applications. Understanding DNA replication is not just fascinating, but it also has practical applications. It helps us study genetic disorders, develop targeted treatments, and use techniques like PCR, polymerase chain reaction, for diagnosis. Conclusion. To sum up, DNA structure and replication are vital in genetics and biology. We have explored the twisted ladder shape, how the strands run in opposite directions, and the three replication steps. By understanding these basics, we gain insights into the incredible world of DNA. Thanks for watching.